I've got a series of questions we can work through, but I really would like to hear from the audience in this. So those who've got a question, please put up your hand and we'll get some mics out to you. And while you're doing that, we will deal with one of our first questions. I don't know if we got, but there's just a person over here. So Ash, great, grab my mic down there too. Okay, so why we're getting organised. So obviously regulatory is an important part. So I'll just get to have Colin and David just comment on the regulatory approach that they're following. So Colin, do you want to kick us off?
Yeah, we're going to go where it's fastest. David and I have something that's unique in what we're doing in terms of developing a vaccine. It's at a different price point in terms of what it costs to do this research than working your way through a feed additive approach. Now, the regulatory pathway and the feed additive approach is not a happy space either, but the beauty of the world is that of the 1.5 billion cattle, 60% of them exist in 3 jurisdictions. And so the number of regulatory approvals we'll actually need to have a massive impact is not high. But getting that first regulatory approval is important. And we heard from, in particular, the New Zealanders and Australians today that there's the ability to shorten path if this is of real importance to that economy. So there are places to move quickly forward. The US, which, obviously we're a US company, but the pathway has become increasingly unclear over the last 6 months or so. So, we will continue to look for that first market that gets us proof that we can get into enough animals to give us all the operational data that we need. Again, phenotypes are important. Carbon and methane mitigation is what we're here to do. No one's going to believe it unless we can measure it, so we have to be in a situation where we can bring measurement, rapid vaccine development and rapid regulatory approvals together, and that's kind of our focus on that first vaccine, after which there's a limited number of markets that we'd like to get into quickly.
David.
Yeah, thank you, Paul. Thank you, Colin. To complement what Colin outlined on the geographic side, which is also important in our case, just some reflections on the regulatory process. There's obviously some important considerations to balance. We heard from some of the the regulators yesterday. We know it needs to be effective. Farmers need to know how to dose the animals, and we need to be comfortable around the risks, both safety and trade risks, certainly for for export-focused countries. We think there's opportunities, given this is a vaccine not against a pathogen, for us to explore regulatory approaches in the sort of markets that that Colin described, where we can bring vaccines to market that meet those core needs from a regulatory perspective. And also use the deployment process to improve the data through the the large-scale deployment of vaccines in the field to refine how some of the quantitative elements of the regulatory approach look. So we've found the regulators to be both responsive and engaged. I think that's a theme that came through from regulators yesterday. Regulators are hearing from farmers, they're hearing from stakeholders in the ecosystem that farmers, ranchers, they want these tools, they want them to meet the needs of their customers, and so we're really encouraged by the sort of responses that we're getting from exploring the regulatory process so far. Thanks.
Can I just add one more thing, Paul? To the point that John was making in his presentation, the immune system of cattle have seen multiple challenges from methanogens through their life cycle. So those antibodies already exist in the blood of cows, and you can probe for them and find them there. And that way, if you think your typical vaccine, which is bringing a novel challenge to the immune system, we're bringing something that's already seen, which gives us the challenge on how to make sure it's continues to be presented, but it does make for an exceptionally safe vaccine in terms of acceptance and consumer safety.
And just to clarify one of the points that Colin was making, this in the US in particular for vaccines, Normally, animal vaccines, it's all USDA, but there's a slight wrinkle there, and that is that they're vaccines that that manage a disease. Because this is not viewed as a disease, then it's FDA. There's only been one vaccine for veterinary species ever licensed by the USDA. I was involved with that work. That's Improvac, to control testosterone. They didn't view testosterone as a disease, but probably half of the people in this room would. But that's the split between USDA and FDA. But Ash, I think you've got a question.
Thanks, Paul, and thank you everyone on the panel. So if you think back to COVID and how fast we can develop a vaccine if there's the incentives to do so, and then when you look at what happened there was a mixture of competition to get companies to strive and develop faster, and also collaboration where they shared efforts or joint ventures or whatever they happen to be. Where do you see the balance with your vaccine development between that collaboration and the competition?
I might comment to start with, sort of from the Global Methane Hub. So when we had that meeting in Dubai, we looked at the areas outside of the people working on the vaccine, the two companies, was a nice competitive element, is what are the other things? And as you heard from John, one of the logical things was to bring people in who had new ways of looking at. And so one of the early things we did with the support of the Bezos Foundation was to bring John and his team in to actually use these really sophisticated tools to have a look at it. We've also got groups working on the early recognition. Colin's just commented that methanogens are recognised, not particularly strongly. So that's where we brought those groups to sort of do a bit more basic research in that area. But I'll open it up to any of the panel. John, you want to have a go at that?
I can't talk too much to the competition in this field, but I don't think the COVID vaccine example is particularly useful when you think about the speed, because with coronaviruses, we knew what the antigen would be. Vaccines already existed in multiple species for coronaviruses. So it was changing what we already had and putting new technologies. This is a more complicated vaccine challenge.
Yeah, I think just to build on the comments from John and Paul there, it is a more complicated challenge, and we're really keen to learn from the best of what we saw in COVID and other experiences. So we're hearing from a lot of solutions at this conference. We see vaccines as a complementary solution to other tools, to feed additives, to genetics, to improve pastures, to different measures that folks can take. We're really keen to collaborate with folks in the ecosystem. There are things that even Livestock Bio and ArkeaBio, as companies that are both developing a vaccine, can work together on. The early regulatory engagement to, particularly given that it's not a regulatory process to address pathogens. What are those processes that help to streamline the approval for any of the vaccine companies? The collaboration with folks like John and others on the research side. We've got partners from AgResearch and others in the audience. So I think collaboration will be critical, as it will with funders. There is an important monetization piece to particularly the early deployment of the vaccine. Philanthropic foundations are interested in seeing this sort of solution deployed in developing countries. We're seeing collaboration through the value chain. Customers of large dairy food companies working together to provide mechanisms that, for example, in New Zealand are already paying a premium for lower emissions milk because the large customers of our dairy companies want to see those emission reductions in the supply chain. So, there's plenty that we can and will compete on, but we're talking about a massive global challenge, similar levels of annual emissions to the total emissions from passenger cars all around the world, without the tools that farmers need to abate in all the farming systems that we see. So this is a huge global challenge, and collaboration will be really critical to success in this space.
Excellent. More questions from the audience. Yes, down the front here.
Yeah, I'm Tim Hackman, room microbiologist here at UC Davis. The ultimate target of the vaccines are methanogens. How do you deal with the fact that there are so few publicly available isolates of rumen methanogens? I think the DSMZ has 7, and most people just use 1, Methanobrevibacter ruminantium M1. Thank you.
Who wants to take that one?
Sure. So the way we're trying to deal with it is to take isolates that we've received from our colleagues in New Zealand that represent as diverse a cohort as we can, and then we're driving cross-reactive responses across that in a way, in a holistic way, where we're not implying what the antigen should be. We're then going back and looking at what those antibodies are seeing and how representative those antigens would be across the population. But there will be an iterative process about what works and what doesn't.
And we've recognised that as well. And there are some publicly available. We've got isolates of our own that we've taken the time to work with. And so we do have effective in vitro mechanisms to look at methane reduction as well. But to go to John's point, looking for that broad spectrum approach is really important. And in terms of the previous question about competition, the competition here is to find the antigens. And then there will be further IP that's generated across execution in the value chain around holding that longevity of response, increasing the efficacy. But there's multiple points of contact that we can collaborate around on the pre-competitive areas and on the industry good spaces, that rather than having one company forge the way, it becomes much easier to do. And we look to the Global Methane Hub for where those things exist and look at ways to continue to drive speed into market, because as much as anything else, if I've got half the antigens and he's got half the antigens, and we're not going to market because we haven't got there, then we're not doing our shareholders any good anyway. The size of this market and the discounted cash flows we can take, say, it's going to drive innovation and it's going to drive the competitiveness initially, but it's also going to drive partnerships very quickly.
I'd add to that, the beauty of the approach here, where it'll be a synthetic, is you don't actually, as long as you've got a sequence of the methanogen, you're fine, and there are fortunately hundreds of sequences available. So in that regard, it's not as big a disadvantage if we'd tried other approaches to vaccines. Any other questions? Yep.
Thank you all for really interesting talks. My question was around, given that methanogens are not pathogens, if a vaccine were to be successful in either eliminating or reducing the methanogen composition of microbiomes, is it understood which other microbial species are likely to compete to fill that gap, and if that might have effects on other attributes of the animal, for example, in nutrient processing or the like? Thank you.
Anyone want to take that one?
I'll jump in first. This is where we look to some of the work that's been done by our friends in the feed ingredient space, the additive space, who are able to get to high methane mitigation rates through dose response. And we, as much as anything, would love to be able to tell you we could give you an answer to the problem, but until you're at those types of performance, we can't really look to it. So we look to some of the literature that's been developed in that space, and certainly we are consistent, as we see at the levels of mitigation we're at, we don't see any change in DMI and ADG and other attributes around the animal. But we look to the literature at this stage to see that the modification of the microbiome doesn't seem to impact many of those attributes at all.
And I think as for the feed companies, our target is a productivity improvement. Taking that energy that's currently going into that methane formation process and putting it to more productive use, more meat. There is a lot of work that that still needs to be done on that, as Paul mentioned, on that target product profile. It's important that at a minimum we're at least neutral on productivity. And we think there's some really interesting work that, as Colin mentioned, that feed additive companies have done and will continue to do, looking at the interaction between these variables. And there's a lot of expertise in this room around some of the dimensions there. So we think that's a really interesting space to explore. And there's a lot of other things that we need to make sure we solve in parallel.
All right, I think we've got time for one last question if there's one out there. Yes, just down here. Yep.
Thank you for that great presentation and talks. Quick question to John and the other commercial companies. In terms of the time frame or duration when you expect to have the vaccine ready, because according to John, there's some complexity, it's not very straightforward. But commercial companies always have commercial interest. So can you tell us maybe when should we think of having a vaccine ready in the market?
It's a great question. It's probably one of the most common questions we get asked. So our current focus is on driving towards proof of concept in cattle. There's 5 key dimensions that we're focusing on beyond that as key components of a commercial vaccine around efficacy, vaccination strategy, safety, affordability, and productivity. Our focus in making that progress towards those key commercial components of a vaccine is to unlock the ability to secure an ongoing license deal with a large animal health company. Back to that commercial question of how do we accelerate time to market. We have a licensing business model, and very much our goal is to get vaccines into the hands of farmers working with our licensed partners within the next 5 years, and we're driving towards that. So big prize, a few things to work through between now and then, but we're really excited about both the near term and that ability to get tools into the hands of farmers.
Yeah, I think, as a person who's worked in the vaccine industry, generally once we have proof of concept, it's a 3 to 5 year horizon to registration, depends on the market. Longer in the US, obviously, for this one with FDA. But I think that's quite feasible. I mean, there's a lot of challenges here to find the right antigens, but there's nothing particularly unusual in the type of vaccine that would be made. We make synthetic protein vaccines, all of that piece of technology is around. So there's no hurdles there. The big here is getting a good solid proof of concept. Colin, I don't know if you wanted to add anything?
Yeah, I hope there's no VCs in the room that are listening to you say 5 to 7 years. We think it can be much quicker than that. And a lot of that depends on the business model. It depends on how it's going to be monetized. And so 30% is 30% because, well, everyone else is at 30%. You don't need to be at 30% if your MRV, if your measurement, recording and verification is done inexpensively. So finding ways to drive cost out of the verification side and finding ways to get into animals with low customer uptake costs is going to be key. So we think that you can be below 30% and make a significant impact, certainly in an initial rollout. And we think that regulatory timelines can drop to half to a third of what potentially they are in the standard vaccines in those markets that have a real focus on getting there. So again, we want to make sure that we are in market in a way that can make a noticeable difference coming into the 2030 timeline when people want to be hitting those Scope 3 or meeting those Scope 3 commitments.
Just to add, the complexity I'm talking about is running in parallel with the efforts that these guys are doing. It shouldn't hold up a vaccine, but we'd be foolish not to use the opportunity and the funding and the momentum to already have in line improvement, second-gen vaccines, how these work. And that's really where the synergistic approach, I think, between us is important.
So I think we're out of time now, but there are people around, so please catch them in the breaks. Just leaves us to thank the panelists, I think, for a really interesting session today.